session 12, site session 12 for this IOC-WESTPAC kickoff conference for UN decade. So I will start first, Kazumi. Oh, okay. Yeah. Okay. So first of all, I would like to thank everyone, all the speakers. Elsa, please make sure that your mic is muted for the moment. Then when you want to speak, then you can turn on and raise your hand. Use the raise hand function. And then I think the secretariat actually asked us to have a group photo. So I would like to ask everyone to turn on your video now. So then we can start with the group photo. And I guess this is a very important business for record purpose. So we probably try to do this first and get everyone to have, the admin will help us to record the whole meeting will be recorded, and eventually the recording will be shared online. So please take note of that. I think the recording already started. And I guess now we can have our group photo. So please, admin. So because there's involved more than one, there are more people, so it will probably take some time. So admin, please let us know, and so when it's ready. Okay, so I guess
Miss Tan, I already okay, now it's better. All right, so everybody ready? And some people they don't have camera on their computer, which is fine. So 1, 2, 3. Just a moment. One more shot. Okay. Someone just come.
Okay. If that is ready, then we can. Okay. So
give me a minute. Somebody trying to turn on the camera.
Okay. Yes.
Miss Riani, I only see half of your face. If you could move back a little.
Please keep your mic muted if you are not. So to ensure that we are able to go through the presentation and discussions later on. Yeah. Thank you.
All right, that's all. Thank you very much.
Okay, thank you very much, Amin. So I think we have to start with the agenda today. So first of all, I would like to thank everyone for coming and joining our sessions. And this is an event that organized initiated by IOC-WESTPAC Hub program. So, and there are two conveners for these sessions, Dr. Kazumi Wakita from Tokai University and myself from Potin Lim from University of Malaya. So I will give some brief intros on the rationales and the objective. We are very happy to see Fukuya Sensei here with us. So I guess that will be, we will certainly get very useful feedback and input from him. So as the founder of this program. So let me share my screen and a few slides that I have for today. So, okay, I hope you can see. Are you seeing my slide now? I also got confused. Okay, so are you seeing the slide now? So the title of the sessions is actually fostering transformative hub sciences for societal applications. I think we are aware of the UN Decade Oceans and UN Decade for 2030, and because of that, and because of the importance of harmful algal bloom, and how we need to do what we need to do to align with some of the UN Decade Ocean outcome. So, and that is the purpose for the meeting today and also the discussion today. And we have identified several speakers that are trying to, give us some idea and some advancements that we have in HAB sciences that might be useful to aid our discussion for today. So I guess for those not familiar with the 7 ocean decadal outcome, safe oceans, productive oceans, predictive oceans, those ocean outcome that we want to achieve that very much related to harmful algal bloom, the importance of food safety, food security, and so on. So with that in background, so, and I guess we are all aware of the WESTPAC HAB program that have been there for so long, founded by Professor Fukuyo. So, and it's time for us to look at what we have achieved so far, and also what we need to do to move forward. And we have several speakers. I would try go through the introductions, and we will start with the talk by Dr. Iwataki, Mitsuru Iwataki from University of Tokyo, to look at the HAB species that we have, both biology and ecology, the identifications of HAB in this region, what are the emerging species that we should look at, and I guess he will give us some idea on for further discussion. So I think this slide. So Dr. Shi Kensun from will talk about metabarcoding and DNA analysis, and also Kazumi Wakita from Dr. Wakita from Tokai University will talk about the socio-economic impact of HAB in this region. Then followed by Aleta to look at modeling, so which is quite modeling and early warning, which is very, very important. And then we will have 2 presentations from Korea and China to look at the HAB monitoring program. And at the end of these sessions, we have a discussion. So I try to keep some time for discussion. I guess that is very, very important for this particular meeting, what we need to do to come up with some plans, some activities that can contribute to UN Decade. So that's why it's a Decade Actions Incubators. We hope that we can come up with something to reach that. So and in the process of applying for this the kickoff sessions, we also apply to get global HAB endorsement. So we see here what is global HAB. If you like to know more, this is a international program that's supported by IOC and SCORE at the same time, and there are it's a continuation of program from global HAB, JOHAB to global HAB now. And we also have a documentations related to JOHAB Asia that will be very, very valuable document during Fukuyo's time and Professor Furuya's time. So those are the documentations that we should refer to look at what we want to proceed and how we want to proceed from there. So I guess the issues of HAB is very complicated in these regions, and we have to look at various type of issues, not only the land-derived input, but also how the changes, not only the surface, but also the bottom, not only the fish, but also shellfish. And we are seeing more shellfish and other marine invertebrate that have been impacted, like what happens in Hokkaido recently, the Carenia bloom that related to the massive mortality and fishery of sea urchin. So, and I already mentioned about the 5 speakers that we're going, 6 speakers that we have here. So I guess we will go through, we'll start with talk by Dr. Iwataki. And the time given is 12 minutes, even though we stated 15 minutes here. I hope that we have 3 minutes, roughly about 2-3 minutes each, if possible. But at the end of the sessions, we have about 25 minutes, because when I followed the sessions yesterday, we noticed that there are many sessions that there's no time remain for any further discussions among the the speaker and also the participant. So I guess that is something we would like to achieve. So I think while looking at that, try to think about the what, the where, the how, and the when, why, and who about hubs. So I guess these are some of the questions mark that we might have related to maybe that you can post to the speaker later on about this, and what then consider this in your in our discussions at the end of the presentations. Okay, so without further ado, so I would like to invite Dr. Iwataki. So the time is yours. Thank you.
Thank you, Botin. Can I start?
Sure, sure.
Yeah. So I think my face is not necessary. I'm Mitsunori Iwataki from the University of Tokyo, working on the taxonomy and phylogeny of microalgae, especially the marine harmful dinoflagellates. In this, my presentation, I will introduce the causative species of Habs. Oh, yeah. How to go ahead? This one? Oh, yeah, okay. Harmful algal blooms have a natural phenomena of proliferation of the microalgal or macroalgal species causing problems on fisheries and human health. Fisheries damage of Habs are caused by harmful algal species, which kills marine organisms such as fish or shellfish, and human health problems of Habs are caused by toxins, which is accumulated in shellfishes or fish that consumed by humans. These harmful and toxic effects are caused by microalgae, and we are studying to identify such causative species of Habs. So to understand the proliferation of microalgae and to mitigate its negative effects, monitoring of Habs are carried out. Basic skills of Hab monitoring is microscopic observation to detect harmful algal bloom species and to count the cell number to understand the increase and decrease of cell densities of Hab species. For example, in this light microscopic field, or we usually observe like this, the many long and brownish cells are thicket dinoflagellate celatium. Some other transparent or slightly reddish protoperitoneum, like this. Brownish diatoms, like this. And small numbers of DST, diuretic shellfish toxin producer, Dinophysis species indicated in red circles, like this. So the identification of the Habs are basically depending on the skills and knowledge of Hab species. For microscopic identification of the Hab species, harmful red tide and shellfish poisoning causative microalgae are understood. Actually, these slides are prepared by Dr. Fukuyo. For example, harmful red tide species of the dinoflagellate Cochrodium polycricoides and Karenia mikimotoi, and PSP, DSP, ASP causative species such as the dinoflagellate Alexandrium dinophysis and the diatom pseudonychia. In the global scale, available information of the Hab species have been compiled at the IOC-UNESCO taxonomic reference list of microalgae. And all we can access to the information of all Hab species in this webpage. Please visit. This Hab species information is managed by taxonomy task team members of Intergovernmental Panel of Harmful Algal Blooms. We had a workshop last week, and we report the updated information of the relevant taxonomic group, such as new paper references, distribution, and the DNA sequences of each Hab species until the next workshop in the next March. In this IOC-UNESCO taxonomic reference list of microalgae, information of Hab species have been updated. Currently, almost 200 species in total, which consists of 45 cyanobacteria, 31 diatoms, 8 haptophytes, Dinoflagellates are separated into lower taxa, such as for Amphidinares, 12 Dinophysiales, 47 Goniolacares, 19 Gymnodiniares, 2 Peridiniares, 13 Procentrales, 3 Toracospirales, 4 Amphidomataceae, and 6 Rhaftophytes, 3 Dictyocophytes, and 2 Pelagophytes. Of these, 87 species have been listed after the year 2000, including new species and new combinations, and 43 species after 2010. Like this, HAB species in the world list is still increasing. In the Western Pacific, this region, we also know the HAB species occurred in this region. Shellfish poisoning toxin producing species like the Parodinium bahamense, Alexandrium species, Pseudonychia species, and harmful red-tide species such as Cochlodinium polycricoides, Karenia mikimotoi, and Chatonnelle species are common in this region. Now I talk about harmful species, harmful bloom species, and its local list. This is an example from Nagasaki Prefecture, Japan. Harmful blooms are common in Nagasaki, and the list of causative species with their cell density is prepared for HAB monitoring. For example, 1 cell of Chatonnelle antica and 100 cells of Karenia mikimotoi per ml are detected. Then information is provided to relevant community and people, such as fishermen. If the target species is recognized in the HAB monitoring area, it is helpful for the early detection and quick action for mitigation of fisheries damage. In Southeast Asia, fisheries damage has so far been caused by Rhaftophyte Chatonnelle species and Dinoflagellate Cochlodinium polycricoides, Karenia, Callodinium, and Takayama species. This information is given by Iniges et al. This year. She is actually a speaker of this session. In East Asia, major harmful algal bloom species are Rhaftophytes, Chatonnelle species, and Dinoflagellate Karenia mikimotoi, Heterocarpus circular squama, Cochlodinium polycricoides. Information of causative species and fisheries damage are provided in Sakamoto et al. This is also published in this year. For preparation of local HAB species list, information of species distribution is useful. However, detailed information is limited in this region. This is some example for species of Chatonnelle, Cochlodinium, and the Kareniaceae. Recently, distribution of HAB species is compiled for each species in Chinese course by Gu et al. This information is also very useful. As for the Rhaftophyte Chatonnelle species, distribution information is compiled recently by Lam et al. In this year. It had been reported only 3 cases until 1989, and now wide distribution of this species is recognized and in the species level with the help of the molecular markers. In the Western Pacific, the HAB species have newly been detected recently. For example, fisheries damage was caused by Callodinium australis in Malaysia and Singapore in 2014 and 15. Takayama species in the Philippines in the 2016 and the year after. Karenia species in Kamchatka, Russia in the last year. Also now, severe fisheries damage such as mass mortality of sea urchin being caused by the dinoflagellate Karenia seriformis in the Pacific coast of eastern Hokkaido, Japan. This harmful bloom started early in this September and now still blooming. The bloom of Karenia seriformis is the first recorded in Japan, and the fisheries damage would be the worst record in terms of the economic loss in Japan. Therefore, we also need to keep updating the HAB species list in the local and in this region. So for summary of my talk, number of HAB species are increasing in the IOC UNESCO reference list of harmful microalgae due to the reports of the new species and new combination. Number of HAB species are increasing also in the WESTPAC region due to the new records of HAB species in this region. Local HAB species list is helpful for the local HAB monitoring, and the regional HAB species distribution can be utilized to prepare local HAB species list. Thank you for your attention. Oh, yeah.
Yeah, is there any questions for Dr. Iwataki? I didn't see any raising hand raised for this, but probably start with me. I think we have a bit of time to ask. You see, consider Japan has probably one of the most comprehensive monitoring program on HAB, and with very long history and record. And the occurrence of this Carenia saliformis is something not in the list, or actually a new record in Japan. And is that any effort to improve in terms of detection of this cryptic or rare species in our in our own coastal waters? I guess that is the questions that I have for you.
Yeah, thank you for your question. Actually, the occurrence of this species is the first record in Japan and probably also in this region. But last year, the same species was detected from Kamchatka, Russia. So in Japan, we now try to obtain the many information about this species. For example, the cell shape, because the cell shape is actually a bit different from the original description that was given from the New Zealand, and after that reported from the Chile. So now, we try to compare that species, and maybe later we report the occurrence and the morphological and the phylogenetic information of the Carenia saliformis.
A subsequent follow-up questions on this is since you mentioned about the occurrence of bloom actually started in September, is there any effort by the locals or the fishery authority to mitigate this particular bloom, because the scale seems to be huge for our large area? So but anyway, so any information on that?
How
like
to answer?
Modified clay, or the removal, early harvesting, so to minimize the impact. Is there any effort that we can learn from that?
This time the fisheries damage is the bit different from the usually we have in the US and Japan, because usually we have the fish kill of the farmed fish. So we try to mitigate the damages to the early marketing or the some other methods. But in this case, the sea urchin were killed, but we just noticed after the mass mortality. Probably that was already killed 1 week before. Like that. So difficult to mitigate that. And also the fish mortality, that is something like the wild salmon just catched by the fixed net. So we cannot manage that fish kill.
I think you, Dr. Yu, from you have questions, and Nguyen maybe follow up with that. So Dr. Yu.
Oh yes, thank you. Just a question follow on your questions about the Carenia saliformis. So last year it formed a very intensive bloom in the coast water of Russia, and this year in Japan too. Just wonder, Professor Iwataki, could you give some idea on the origin or the mechanism why this kind of bloom will happen?
Yeah, thank you for your question, Ren Cheng.
Yeah.
Yeah, actually, we are now thinking about that mechanisms. Yeah. Actually, that similar bloom was detected in Russia last year. So if the population is the same, that red tide or the seed population what came, but that area is very cold. We still don't know how to overwinter, how to survive under the very low seawater temperature. Now we try to obtain that kind of information. For example, around the sea surface, during winter time, that's around 0 degrees Celsius. And also in the 100-meter deep, that temperature also around 0. So yeah, at the moment,
a lot
very
of
difficult to
interesting
consider.
questions.
Yeah.
Yeah.
Okay, I think when you have questions that.
Yeah, I'm interested in the sea urchin diet in Hokkaido. Is it the culture or the natural sea urchin? And what is the mechanism of killing? It's depletion, oxygen depletion, or toxin, or whatever, because it's quite interesting and new to me. Thank you.
Thank you, Nguyen.
Yeah.
So I can answer. We don't know.
That is
but
true.
probably
Okay.
that species produces some kind of the bioactive compounds that affect to the sea urchin. But we still don't know what kind of the compounds produced by that species, and probably not by the oxygen depletion. Yeah, because that area is not closed.
Is that natural sea urchin?
Yeah. So how to answer that? It's actually that juvenile were released, but something like a natural.
Oh,
yeah. Not
okay.
in the
I think we can, if you still have questions, we can keep it during the question and answer sessions. Then we move on with at the next speaker, Kian Sun, Dr. He from University of Malaya. So yeah, you can share your slides.
So okay. Okay, can I start now?
Yes.
Yeah. Okay, good morning to everyone. So my name is Kin Sun from University of Malaya, Malaysia. And it is my distinct privilege here to share with you all about the recent advance in the harmful algae bloom with as the title harmful microalgae monitoring assessment with the meta-barcoding approach. So the rapid developments are subjected to the disturbance from the land debris or other anthropogenic activity. They lead to the deterioration of water quality, and nutrients enrichment, or also known as eutrophications in the water, have driven the shift of the phytoplankton community from the high species diversity to monospecific. They often form the harmful algae bloom, and it has caused the fish kill and seafood contamination due to the microalgae biotoxin. And beside that, the high biomass algae bloom due to the high nutrient will also lead to the dissolved oxygen depletion in the water. They cause the fish die in suffocation. So the impact of the harmful algae bloom and algae bloom has been well recognized. However, the fundamental research effort on the mitigation, detection, and prediction are still less. Therefore, the effective the HAB management and mitigation effort are crucial, which is relied on fast, reliable, and accurate tools for detecting or monitoring the HAB species. So as you all know, the traditional conventional approach such as classical taxonomy identification with the microscopic method are time-consuming and labor-intensive. So the microscopic approach also have a higher degree of the inconsistency in identifying species, and this becomes further complicated owing to the co-occurrence of morphological closely related species or critic species complex, such as the Alexandrian species or Pseudonesia. And also some fragile species, such as the naked dinoflagelate and the Rhaphidophite, are easily deformed morphologically or burst during the sample handling that make the microscopic enumeration become difficult. And the microscopic approach also required with the taxonomy expertise or certain skill in species identification, while the identification mostly only until the genus levels. Therefore, the highly accurate genetic distinction approach, which is the meta-barcoding, are essential to monitor the HAB species. And meta-barcoding is a fast genetic approach to elucidate the comprehensive taxonomic profile in the environment, and it is reliable due to the taxonomy assignment are based on the sequence similarity on the reference genome in the huge database. So compare with the traditional method of the microscopic approach, with the meta-barcoding, the large number of samples can be processed simultaneously, and then of course it can reduce the cost associated with the method, and also the turnaround time of getting result is reduced, and also the sensitivity and the specificity is quite enhanced, which it can capability to detect previously unknown species or novel species. So as you can see below here are the workflow of the meta-barcoding. So the meta-barcoding start with the collection of the environment sample. So it could be the water samples, sediments, or soil. So for the water sample collection, generally the environmental plankton can be either collected by using the submersible pump, bentons or plankton net. While the sample volume, for the sample volume, the collection water sample volume is based on the environment. For instance, for the offshore, usually can be collected more than 10 liters, and for the coastal, it can be within 1 liter or sometimes can be more. And the collected sample need to be filter fractioning based on the desired targeted cell size and process either with the saline ethanol or formaldehyde, then followed by store the preset sample in 4 Celsius or -20 Celsius to prevent the DNA degradations. So the environment sample were later brought to the lab where the eDNA of sample were extracted, and this followed by the PCR amplification of one or several taxonomy informative marker or primer. They're able to catch the entire examples, for example, bacteria, eukaryote, or planktons. And so the perfect meta-barcoding DNA should be present in all the organisms or cells. And the sequence of the target regions need to be variable among different species and constant among the individual of the same species. And it also need to be easy to amplify and not too long for sequencing. And of course, the sequence region need to be available in the accessible database reference such as the GenBank, PR2, or SIBA. So as you can see, the table here are the common meta-barcoding marker they use based on the different target organisms. So then the umbilical of the targeted region will sequence with the high throughput sequencing technology. Even though nowadays the new sequencing technology continues to emerge, but the Illumina MindSeq currently dominate and has smoothly replaced the old technology, which is the ROM 454 sequencer. Then followed by the bioinformatic processing and the species identification. The read processing and the bioinformatic is usually done by the package such as the Chime tool, Mortar, or Dada tool. And the sequence read generally will undergo with the read pairing and demultiplex, removal or correction of sequence error, removal of the chimeras, primer treatment, clustering and replications, and taxonomy assignment based on the reference database such as the GenBank, PR2, or SIBA to generate the operation taxonomy unit, which is the OTU, or variants of the identified taxa. Then the rate of the representative OTUs or variants of the TASA can be further used in the ecology analysis, such as the diversity, community structure, and population dynamic. So in here, I would like to show you the study from my group. They use the meta-barcoding approach to investigate the temporal partial change in the harmful algae bloom community example along the Johor Strait. And this study not only show the sensitive and the reliable of the meta-barcoding in the HAB species detection, but it's also useful in the harmful algae bloom community example dynamics. So for your information, the Johor Strait is an important site of the marine culture for Malaysia and Singapore. Besides the marine culture activity, the strait also experiencing the huge ecosystem damage due to the rapid economy development involving the various human activities such as the seafloor dredging for shipping lane maintenance, land reclamations for the resident or the commercial properties. And from this study, the eDNA of plankton sample were collected monthly between May 2018 and September 2019, covering with the 90 stations across the Johor Strait. So while for the methodology of this study, the plankton sample were collected using 50 micron mass plankton net followed by a vacuum filter with 0.2 pore size of the nylon membrane, filter and preserved in a saline ethanol. And the eDNA of the plankton were isolated by using the commercial extraction kit. And the V918 srDNA was amplified by using a pair of specific eukaryote primer. And then the umbilical were analyzed by the Illumina MySeq, and the bioinformatic processing was done by the MOTA. So from the high throughput sequencing meta-barcoding data, a total of 377 phytoplankton TASA were reviewed from the 170,000 breed, and several predominant items such as Sclerotinia, Eucampia, Rhizosilenia, and Thalassosilla were relatively high in the data. So from the alpha diversity analysis based on the OTU risk of the phytoplankton TASA, we found the higher phytoplankton diversity were observed in the outermost station of the Johor Strait. Then beside that, with the high sensitivity and the specificity of the meta-barcoding approach, we can manage to discover 26 OTU was associated with the harmful algae bloom species, where 11 OTU of the HAB species belong to the previously unrecorded species in the Johor Strait. So we also can find that the highest HAB species richness were observed in the outermost station, and the OTU of the HAB species were further analyzed by the dual hierarchical clustering analysis to investigate the distribution and abundance of the HAB species in the Johor Strait. And from the analysis, we show that the Heterosema akashiwo, Verbukasa japonica, and Pseudonesia panjang were distributed in high abundance throughout the Johor Strait. And with the Venn diagram analysis, it showed that the Pseudonesia pseudodelicatissima was only detected in the western Johor Strait, and for the Pyreneans and Chatanella sasansa, Caledonian beneficent, and the Poricula hemani were only detected in the eastern Johor Strait. And beside that, the OTU of the HAB species can also correlated with the ecology parameter such as the temperature, pH salinity, dissolved oxygen, and variant nutrient constraint to assess the characteristic or trend of the east HAB species examples. So for instance, in here, the CCA show that the Pseudonesia delicatissima, Pseudonesia multiciliata, Chatanella mariana, and Verbukasa japonica were exhibited in the ambient with the tight mixed mass, which is a high turbulence, low temperature, and relative low MP ratio environments. So if you're interested to know more about the finding funding of this finding of this study, you can find the article from this article from out there, harmful out there. And as the conclusion, metabarcoding approach is useful for the HAB species diagnosis due to its sensitivity and high accurate detection. And the information of the HAB metabarcoding sample across the potential hotspot will be useful in identifying the source of the bloom and possibly predict the probability of bloom. And the metabarcoding data provide variable baseline data of the marine phytoplankton and HAB species community example diversity. And it will be crucial for future monitoring and study related to climate change impact on the coastal productivity, ecosystem health, and food resource in the coastal water. So, and thank you, and I'm happy to take any question from you all.
Okay, thank you, Kenson. So, if there is any questions from for Kenson, please raise your hand. Then we can have one or two. Yes, maybe start with Dr. Iwataki. Then after that with Dr. Wang, hoping. Yeah. So.
Thank you, Kenson.
Yeah.
For the interesting talk. I'm interested in the molecular species. Previously, I think the SSU, ATS, ribosomal DNA sequences were widely used, also for the metabarcoding. But currently, the ITS or the LSU, the reference data is deposited, I think, mainly for dinoflagellate. How to arrange that kind of the molecular species to detect the harmful algae? In particular, the dinoflagellates.
Yeah, this is something related to the reference database. So in terms of, nowadays, even though we have a lot of reference database at NCBI, PR2, and SIVA, but all of them data is actually not robust enough. So that means some species will not have the sequence inside the database. So this is become something a challenge for us for doing the metabarcoding. So the one thing we can do now is whether we can establish. I hope we can, or in future we can establish a complete more robust database for detecting all the HAB species. And I know some species they are more suitable use ITS to distinguish to differentiate or distinguish the species. That's why we need to come back to the database as well. So let's say ITS, we hope that we can have the database is all ITS with the complete HAB species. Then we easily can use the ITS database to identify the species. But even though a different kind of species, they have the different regions to detect it, to identify, to distinguish. But we also can use a different kind of primer marker for detection or to do the metabarcoding in the further analysis. Yeah.
Thank you, Kenson. Peng Ping, maybe a short one. Yes.
Yeah. Can you hear me? It's okay.
Yes.
Okay. Yeah. Thank you, Kenson. It's very interesting talk. About your. Actually, I have some question about the metabarcoding. Just also like the Iwataki-san have referred, no matter how the target regions, and also I also see the data you didn't refer many of the absolutely abundance of the species, right? Of different years. Actually, I want to just if you have see some phenomena of the different sites, as we know, especially for the dinoflagellates, the copy numbers of the DNA, the target regions will be different from species to species, even in the same genus, I think. So before I also do some of this work, but though we can get the very, as you referred, we have it's very have many advantages about the metabarcoding, but I think we still have a long way to go to give some there have many gaps to make real for calculating the numbers of the different species in one times of the right. Do you have any suggestion or comments on these? Because as we really I really find some real case in if we really want to apply these techniques to real monitoring or detecting the harmful algal blooms. Though, of course, when harmful algal blooms, we can know the dominant species. Yeah. But it's for general monitorings, the different species, maybe for example, A and B, maybe actually the real numbers of A is much more than B, but when we calculate when we get the data from the metabolic coding, maybe B is much more than A. Do you know what I mean, right?
Yeah, I understand. This is also one of the challenges that we face in the metabolic coding. We use the metabolic coding, but instead, let's say, we know that the different species, they have the different kind of genomics, copy numbers, size. But in terms of nowadays, I thinking we're planning, I think my junior also, they are trying to do something, we are trying to do something like machine learning. So we try to collect as much sample, then we try to analyze as much sample we have. So from the OTU number we get, we try to use and of course we also want to compare the try to do the comparison with the actual cell number count. So we try to establish a database to do some machine learning, so that from the machine learning we try to correlate it, so that of course machine learning we need to have a lot of data, a bunch of data, so that it can support it and support with the, let's say this OTU, 3 OTU we can representative 10 species, 10 cell. So with a lot of the data we have, let's say maybe 1,000, I'm not sure how much, so that the machine learning can help you to predict or can help you to generate or forecast the data, so that it can show you the OTU can relate it with the cell numbers. And
I think it's still at the very early stage now.
Yeah, so, it's still in a very early stage.
Okay, I think it's almost time. So I guess question from Pumping and Ms. Nori, I guess you can refer to the paper, the HAB species in Gulf of Thailand by Dr. Goose and his group, that related to how to use 3 different markers, the SSU, the LSU, and the ITS. For pseudonyxia, it's only possible to use ITS region. And because we have the in-house database for pseudonyxia, so that's why we can ID all the pseudonyxia in Gulf of Thailand to species level. I get that is some advantage that we have, and that works for some species, some group or some genus. But we need time to develop for Heterocapsa, for examples, for Carenia, for examples. I think that is a lot of effort needed in the ongoing effort that we have to do in order for us to ID to species level. Yeah, I think we have to move on. So next, I think we have Kazumi, right? So yeah, go ahead.
So thank you very much, Potin. So let me introduce my slides. Okay, slideshow. Is it okay, or presentation mode?
Yes, now it's okay.
Okay, so thank you very much. So I'm Kazumi Wakita from Tokai University, Japan. My talk will introduce some recent social science studies on HAB, through which aiming at stimulating your thinking about how social science and natural science can work together to mitigate the negative impact of HAB. So this is a newspaper reporting fisheries damage by red tide in East Hokkaido, as Iwataki-san mentioned. It reported that fisheries damage surpassed more than 4 billion, and now it is set as over 8 billion. So it has become the largest fisheries damage in Japan throughout like over 50 years. So as you know, there are mainly two aspects of HABs, namely red tide and shellfish toxin. And respective impacts of these two phenomena to the society are very different. So for example, when we do monetary valuation of direct damage, so regarding red tide, we usually calculate the dead fish, the economic damage of dead fish. But regarding the shellfish toxin, since the shellfish does not die because of the HAB, so usually what we calculate is about the damage to the human health. So for example, like the hospitalization of the people. And also regarding the impact on communities, for example, livelihoods and cultures. So regarding red tide, usually we calculate the impact on the fishermen and fishery communities. But on the other hand, regarding the shellfish toxin, we have the recreational activity called like clam picking in some countries. So the shellfish toxin also does affect to the citizens for the recreation people. And for us, for the mitigation and adaptation, so for example, clay spray are conducted or to the red tides, to the red tide, some species, and also for the shellfish toxin, some aquaculturists ship earlier than they planned. So what they calculate is that like what will be the economic loss if they did ship the shellfish to the market at the expected time if without a shellfish toxin occurred. And in addition to these aspects, we have also calculation or the studies about the reputation damage and also social acceptance of mitigation and adaptation countermeasures by the people. So to date, a variety of studies on monetary valuation of impact caused by HABs accumulated. And last year, a compendium of case studies on evaluating, reducing, and mitigating the cost of HABs was published, and impact on these aspects are examined. And from our region, Lim and others did a comprehensive study on economic impacts and management of coclodynium in Korea. So if you are interested, please check the publication. And now let me talk about an example of adaptation to paralytic shellfish toxin PST at recreational clam picking parks in Osaka, Japan. This drawing shows a scenery of recreational clam picking called shio-higari around 200 years ago. The shio-higari is a traditional marine recreation and people enjoy it as a family event even today. However, along Osaka Bay, which is eastern part of Seto Inland Sea in Japan, paralytic shellfish toxin occurs almost every year since 2002, and the main causative species is Alexandrine tamarense. In Japan, we have recreational clam picking parks. People pay picking fee at an entrance and enjoy clam picking, and bring clams back home, which they picked by themselves. And operators of the parks, usually fishermen cooperatives, they scatter clams in the area to secure enough clams to be picked. And why is scattering clam necessary? Is because catch of wild clam in Japan, shown as green bars, has decreased. And especially in Osaka, in blue line, wild clam catch rapidly decreased during 1950s and has not recovered since then. In general, if shellfish toxin surpasses regulatory limit by test, clam picking parks are requested to close by local governments. But in Osaka, they keep opening parks even when it is over regulatory limit by taking food safety measures, which is different from other areas. This figure shows system of exchanging clams which secure food safety at a clam picking park. They purchase clams with results of toxin test, and the clams are weighed and packed for exchanging them from those collected by visitors. And surprisingly, this system had been introduced since before the first PSD surpassing the regulatory limit. Why? Because the clamming park is within artificial beach where wild clams cannot grow now. And this was lucky for them to address shellfish toxin. They could adapt to the shellfish toxin through operating this system. And this system from one park has spread to other 2 clam picking parks in Osaka Prefecture. And after the first PSD detection in 2002, the number of visitors dropped down to less than 30,000. But after introducing that exchanging system of clams introduced to all parks in Osaka Prefecture, then the number of visitors recovered to over 50,000. And if you're interested in more details are in Harmful Algae News number 67, so please check it. So if we look back to the WESTPAC region, there has been PSP cases. These are some examples shared among participants when we had a regional training course in 2017 on mitigation and management of Habs. As you see, in some places, PSP occurs and even some fatalities happen, unfortunately. One example happened outside monitoring area, and the other happened through eating green mussels, not through the market, but outside the market. That is through sharing seafood among neighbors. So from these experiences, we can consider how can we do to decrease poisoning cases outside the monitoring area, and how can we design the monitoring areas and systems, and also what can we do to decrease poisoning cases happen outside market. These aspects are some examples of what social science studies could contribute to together with natural science. And in addition to address shellfish toxin, we also need to address red tides, which have geographically expanded and increased fisheries damages. This figure shows trends of red tides caused by Chatonella spp and associated fisheries damage over the past 50 years in the region. To mitigate negative impact caused by red tides, we have many good examples or practices in the region, which will be introduced to later by colleagues. And to mitigate negative impact caused by shellfish toxin and red tides, we can accumulate case studies on good examples and good practices through which we can learn each other and think about what we can do at our respective places in each country. And it will be useful to clarify factors and social systems which enable mitigation, management, and adaptation measures to be successful. As we all know, we have diverse political systems, social structures, economic conditions, and local cultures and habits in the region. So we cannot simply copy one good practice to others, but we can learn important essences which could be applied to our own respective societies through accumulating variety of case studies, I think. So in thinking about Habs, there are three major stakeholders, namely citizen, fisherman, or operator of recreational activities, and national and local government. And this holistic approach as a whole system is important for social science studies and Habs to contribute to address HAB issues. Thank you for your attention.
So thank you, Kazumi. So I would like to invite questions for if there's any. So please raise your hand and then turn on your video so then we can have some discussion on this topic. I think this is very important, and also, and of course, the natures of and background social system are very different. So I guess that is there are lessons that we can learn from this the business model of this clam picking in Japan, and how we can actually, you know, depurate the toxins, which is actually proven scientifically, and keep the toxic clam into clean waters, let it depurate for some time, then we can actually turn it into a different business model like what we have now. So of course, it's shorten time, the need for depurations, like if they keep them into the bloom waters. So any questions from the audience? If yes, Fukuya Sensei, I can see your hand, so please unmute and.
Yeah, thank you very much. Can you hear me?
Yes, yes.
Yeah, I have a question for Wakita-san and Iwataki-san. As Wakita-san shows, Japan has a long history on the mitigation against red tide of the Chatonella. How to save fishermen, coastal society from the damage of Chatonella red tide, because it is one of the biggest fish killer. And is there any effort to apply that mitigation system to the Hokkaido red tide case? Karenia red tide case occurred this year. It killed wild sea urchin and also wild fishes, not only caged, but also wild ones. So if there is any good effort, it will be one of the example. As we have more than 20 years experience of the mitigation against red tide, and I feel it's quite curious now. No report on the effort to avoid or reduce the damage by Karenia seriformis. Thank you very much. If Wakita-san and Iwataki-san knows any information on it, please let me know.
Thank you very much, Fukuyo Sensei. And I am hesitant to answer your question because I don't have good information about what they did about the mitigation. And if Iwataki-san knows, please let me also know, because it is not a good practice. But what I understand now is that in Japan, we have the system of insurance, not by the private sector, but among the fisheries association, fisheries collaboratives. But since the red tide cases only happened in the western part of Japan by now, so the red tide cases cannot cover to that northern area. So that is what I heard about the insurance system. So now they are, I believe that they will consider the geographical expansion of the coverage of the red tide insurance cases. That's all what I know about for the time being.
Thank you very much.
Iwataki-san, any comment from you?
About the mitigation?
Yeah.
In my understanding, we could nothing to decrease the fishery damage in case of the Hokkaido plume. Yeah, all we are now trying to identify and enumerate that just cell number and try to understand what's happening in the eastern part of the Japan.
Maybe some information for me to share. If you follow the early sessions on this Indo-Pacific Convergence Centers, there's one presentation from IOCAS from about this. I think Dr. Yu can talk about more about this, the use of modified clay that they have successfully used it to control various type of bloom along the coast of China, and they also intend to apply and to seek for collaborations with people that are interested in this modified clay. It's already commercially available. So, but of course, that is subject to testing if whether it works, for example, in Philippines or in Malaysia, because the natures of the environment might not be exactly the same. Yeah, Fukui Sensei.
Yeah, also we know some chemicals are very effective, not only clay, but also chemicals such as hydrogen peroxide, spray of the hydrogen peroxide in the wild ocean, open sea, to a certain level of concentration. It kills the phytoplankton without giving any damage to fish larvae. Or some other chemicals such as formalin. It's also useful. But in case of Japan, fisheries agency doesn't like that treatment because public doesn't accept the fish from the area treated by chemical. So it, how to say, public sense without scientific, how to say, true information. But that is the point of the SDG, how to make new technology in considering with the science, social acceptance. Thank you.
Dr. Yu or Dr. Yuan?
Yeah, just followed the comments. I don't know whether Dr. Yuan is in the group or not, but yeah, I think the group in our case has did a very good job in development different types of modified clay. They try to combine the clay with some kind of chemicals to increase the efficiency in removing the harmful algae. But that's not the most important. I think the most important thing is that in the last several years, they have developed several different facilities to try to spray the modified clay into seawater very efficiently. And this kind of technique and products has been used widely in the coast waters of China to treat different types of harmful bloom like Fuchsia globosa, Orococcus anopheles, and this technique was also used to treat long-lasting bloom of Carenia brevis in the Gulf of Mexico in the United States of America. So I think it's a very good choice to to apply this kind of technique for emergency treatment of Habs. Yeah.
Okay, thank you very much, Dr. Yu. I think we can come back to this subject at the end of when we have the discussion sessions. So next we have to move to Aleta. So yeah, so go ahead with your presentations and to see how we move forward with modeling and early detections and so on. Yeah.
Okay, is the slide showing okay?
Yes, yes.
Okay, thank you. Good morning and good afternoon, everyone. So I'll be giving an overview of our work to develop an early warning system for Habs that can hopefully be a pathway towards enhancing the country's capacity to mitigate the impacts of Habs, as well as start for operational ocean observation and forecasting. So as you can see from the long list of authors, this program has entailed the efforts of many people from an intersection of scientific disciplines and organizations, including different universities. I think Dr. Lenny Yap is here, who is also a collaborator, co-author here, and also, of course, importantly, the regulatory government agency. So I'm presenting this on everyone's behalf. Just as a background, the Philippines has been plagued by Habs for, I think, more than 3 decades. And the main Habs syndrome has been paralytic shellfish poisoning, or PSP, and it's been caused mostly by Pyridinium bahamense, with a few sites affected by Alexandrium species that have caused confirmed toxic events. And we've had more than 2,000 cases of PSP through the years, with a few hundred of these leading to fatalities, unfortunately. So the longest record for Habs in the Philippines comes from shellfish bulletins that have been issued by the Bureau of Fisheries and Aquatic Resources. And this represents paralytic shellfish toxin occurrences beyond the regulatory limit. So note that this record predominantly reflects the occurrence of pyridinium in the different areas of the country. And evaluating this historical record showed that in the first 2 decades, there was an increase in frequency and duration of the PST events, most likely due to also increased awareness in monitoring, partly, but has stabilized or even slightly decreased in terms of number and duration in the recent years. But it's still quite a big hazard and a big impact in terms of the number and duration in many areas. Again, based on this record, the occurrence of PST is increasing in terms of the number of sites affected. So it slowed down a bit, but so 1980s to 1990s, on average, 2 new sites were affected per year. But from 2008 to 2017, about 1 new site every 2 years. Although recently there's, I think, a spike in the past few years. So the Philippines has actually an established national HAB monitoring and management program by the mandated government agency BIFAR. And through this system, field monitoring for phytoplankton and shellfish toxicity is conducted in the different HAB-affected areas. And depending on the capacity of the area, samples are either analyzed on site or sent to regional laboratories, or even the main lab in Manila here, to undergo microscopy. And then for the shellfish toxicity, it's mouse bioassay, or at the main lab, they make use of RBA. So these provide information for the issuance at the national level of the Shellfish Advisory Bulletin. And this monitoring and management program has helped to decrease the PSP cases in the country through the years. So of course, there's still room for improvement. There's a few gaps that we wanted to address. It provides now cast only and has no capacity for more early warning or forecasting. And typically fish scales aren't really included in this advisories. It's also only for shellfish toxicity. And there's difficulty actually in collating and analyzing information from the different sites, and including information on water quality, which is quite limited. So manpower, of course, is also quite limited, since we have a lot of areas that are affected, and this can affect the frequency of monitoring and the timely analysis of the samples. So with these in mind, we all collaborated to develop and enhance the monitoring management system for HAB. So to develop this early warning system, which can help address these gaps. So this first component is HAB risk knowledge, which involves understanding the patterns and risk of toxic blooms in fish gills, while the second component is the monitoring and warning service, which involves detecting, of course, and monitoring the HAB organisms and conditions, providing analysis, assessments, and warnings. The third is dissemination communication, involving how best to communicate and disseminate warnings and promote understanding through the different communities. And the last is response capability, which involves how people respond to the warnings or even prior to the occurrence of HAB events, how they can prepare. So I'll go over the key activities as an overview that we've undertaken to build up these different components in our target sites. So for obtaining HAB risk knowledge, it comprised of two main aspects. One is the consolidation and analysis of biophysical information related to HABs, such as phytoplankton abundance, shellfish toxicity, and also the oceanography of the sites. These were through contributions and collation of historical data obtained through research and the agency monitoring efforts. These historical, biological, environmental data are critical in developing any sort of forecast models. So the second aspect is on the socioeconomic information. This was primarily done by conducting a participatory HAB risk assessment with the partner communities at the study sites. So these help to understand the impacts of HABs on these communities and stakeholders, as well as derive local knowledge on HABs. The monitoring and warning related activities can be divided into monitoring on site or the field monitoring, and the other is remote monitoring through various technologies. We had several collaborative field monitoring activities with the various program partners that also helped clarify methodological approaches and allowed for standardization with the university partners. One part of the remote monitoring is the use of ocean color for large-scale assessment. So the semi-automated HAB detection system, or CHABS model, was developed by colleagues Aldwin Almo and Dr. Laura David from UPMSI as well. The model processes chlorophyll A concentrations from Modus Aqua satellite images at 4-kilometer resolution. This was also being developed as kind of an early warning where it can warn of potential blooms using chlorophyll A anomalies. It includes a feedback system called Do You Have It, through which the users, the partners can calibrate, validate, and improve model forecasts by focusing on their sites or zooming in on their sites and then contributing data and information that would modify the anomaly threshold. The second part of remote monitoring is the development of the low-cost sensors for real-time automated water properties measurement. So in collaboration with physicists, and we developed the SensePack, which is a low-cost sensor that measures temperature, salinity, pH, and dissolved oxygen. It is designed to be modular with battery, electronics, and communication and sensor modules, and it can be deployed on surfaces like shellfish farms or fish cages and transmit data at program intervals using long-range radio frequency to a base station. And then the data is stored in a database. Another effort by Drs. Villanoy, Amanda Repollo, and Rachel Francisco also independently developed the automated water quality monitoring system, which is a flow-through setup where water is pumped through the surface through sensors, and data on temperature, salinity, dissolved oxygen, and chlorophyll are transmitted using mobile data. So right now, this is deployed at the intensive mariculture area in Bolinao, Pangasinan, northwest of the Philippines, and it's automated to store the information in the database. So these are still like prototypes, and there's still a lot to improve on for these sensors. At a local site, site-specific early warning models can be used if real-time data is available for a site. And currently a model for Bolinao has been developed, which is a site with the most extensive interannual biological and environmental data set. So this makes use of the random forest algorithm, a machine learning approach, forecasting probability of shellfish toxicity. In this site, it's Alexandrium species that causes toxicity. and also fish kills, and fish kills occur in this site a lot. The model is linked to the real-time sensor data, the sensors I showed on the slide before. So here you see an example of the forecast from July 16, where fish kills medium probability and shellfish toxicity is at low probability. So we're currently observing the actual performance of this system. So essentially what I've shown so far, we have the following technologies in the early warning system with a real-time ocean observation in the field and satellite images fed into the database and informatics system, which also houses historical data from labs and program partners, which are used for then the output analysis and models. And the information are disseminated online and to the relevant agencies and communities. So the system, we call it HAB, the Observation Informatics System. This is the website, and this is a QR code that you can use to go in. Note that it is in beta version currently, and it's literally a hub for the environmental, biological, and model-derived information related to HABs in general water quality. So a key aspect of the program and the development of the system is the engagement of the community and stakeholders from the start of the program. And the participatory HAB risk assessment activity was able to enrich HAB risk knowledge. In itself is a capacity building effort through information sharing and affirming local knowledge and capacities. And it also served as a foundation to develop community actions to help mitigate HAB risks. So from this exercise, the communities themselves came up with some suggested mitigation measures where it's strengthening alternative livelihood options so that they are not as impacted by HAB events, considering HAB as an event that allows for a state of calamity to be called and or having measures for financial assistance during such events. Having a plan similar to disaster preparedness for HABs, such as what we have for earthquakes or tsunamis, and ensuring that communities also have access to information and technologies. So this diagram is actually a proposal on how to integrate these new tools and technologies to enhance HAB monitoring management of the regulatory agency before. The remote monitoring and forecasting capabilities are primarily used to help assess and adjust the needs for field monitoring. And fish kill advisories can be included in local warnings. So the sensor and field monitoring data can be standardized and continuously updated, which can then be, of course, used to further understand HAB dynamics and improve the responses. So the road ahead is still long. Early warning system is essentially in its infancy. And there's a need to strengthen the real-time ocean observation capacity, including, of course, integrating the in a more rapid fashion data on HAB species. So new in situ imaging technologies are available now, but are very expensive and intensive to maintain. We didn't show this here, but we're also exploring the metagenomics, though I think as has been discussed earlier, operationalizing this would be still difficult at the moment. And for toxicity, we're also investigating a modification of the SPAT for toxin detection. Historical data from different sites need to be further analyzed to more concretely establish patterns and thresholds and develop new models. And the flow of processes within the system needs to be improved. Stakeholder engagement and capacities for developing and maintaining the system need to be strengthened. And lastly, and very importantly, in determining how to sustain the collaborations and efforts for the longer term. So with that, thank you very much for your time and to all the support and further collaborators that we've had.
Thank you very much, Anlita. So very informative and very promising future for HAB real-time and early warning system. So I now would like to invite Fuguesensei, you want to have questions? Yes.
Yeah, thank you very much, Anlita. You mentioned in the first or second slide that the area affected by PSP is now still expanding. It means new area, occurrence of PSP at new area. Did you make any effort to understand the mechanism of expansion? How to stop the expansion? Whether it's shellfish transplantation or other natural mechanism? Is there any analysis?
This is a very good question, Professor Fukuyi. We have our hypothesis, which is probably the shellfish transport, because we have had anecdotal reports on transport from HAB affected areas to those areas which have not been affected by HABs and then have occurred. Unfortunately, it's a little sensitive because of the agencies promoting this as well. So this is something that I do want to look into, and also the potential of the cysts to remain in the shellfish and then transfer to those sites and feed the new sites with the blooms. But that is, we would like to explore that further. Thank you.
Yes, go ahead.
Yeah, in BIFA, the first occurrence in Samar Sea, the first occurrence Pyrodium, the people apply the shellfish transplantation project hosted by BIFA, and the shellfish from Kamigi Island, they spread all over the country. Then Pyrodium occurred all around. But still, there is not many study or whether the cyst on Pyrodium attach on the seedlings. And in Japan, we have similar experience. Once in northern Japan, Mutsu Bay, scallop died. The large-scale mortality of scallop occurred. Fishermen wanted to transplant the scallop from Hunka Bay in Hokkaido, where the PSP occurred every year. So the government stopped it and allowed only shellfish seedling, which washed completely using the water, which, how to say, showed UV sanitation system. But actually, by considering the cost, the fishermen in Aomori, Mutsu Bay, stopped the transplantation. So that kind of analysis will be necessary in future to stop the expansion of HAB case. Thank you very much.
Thank you, Professor Fukuyi.
Okay, so I guess we can move on with the next presentations. The next two presentations, we have Dr. Yu from IOCAS, and then followed by Dr. Park from Korea to share about the HAB monitoring in first in China. So go over to you, Dr. Yu. Yeah, please get it into presentations mode.
Okay, thank you, Dr. Lim. Yeah, it's my pleasure to share with the audience here about some of the recent findings on harmful algal blooms related to seafood safety and sustainable aquaculture in China. First, I'd like to start from a short introduction about the status of harmful algal blooms. Here in China, we actually experienced a rapid increase of harmful algal blooms, particularly red tides, from the year 2000. And recently, we can see some decrease in terms of both HAB frequency and the area affected by harmful algal blooms. But whether this trend is real or not, I'm not sure. But there seems to be a decrease of harmful algal blooms in the coast waters of China. So a new phenomenon for harmful algal blooms here in China is the different types of HAB phenomena in different regions. For example, in the Bohai Sea, we have a very extensive brown tides caused by Orococcus and filarians. In the Yellow Sea, we have the green tides caused by green algae our periphery. And in the East China Sea, we're in this area very close to the estuary of Zhanjiang River, we have a large-scale blooms of dinoflagellate. And in the South China Sea, we have the bloom caused by Heptophytes Phyllocystis globosa. And also in different regions, we have different types of toxic algal blooms. And these different types of algal blooms lead to are quite different impacts. The first two impacts are seafood contamination and mass mortality of cultured animals. But besides that, we also have other negative impacts like hypoxia, degraded ecosystem health, and some injuries to the marine layer and tourism, and also the operation of large electricity facilities, particularly caused by Phyllocystis globosa, which can block the cooling system. And there are many agencies in China involved in the monitoring and the studies of harmful algal blooms. We have already established a very systematic monitoring system for harmful algal blooms, which is now operated by the Ministry of Natural Resources. And for the quality of seafood caused by toxin contamination, the monitoring was operated by the Ministry of Agriculture, and they all have their systems for sample collection and analysis. But besides that, we also have many universities and institutes are involved in the studies of harmful algal blooms. For example, the National Marine Environment Monitoring Center in Dalian, and in Qingdao, the Institute of Oceanology, where I come from, are working on harmful algal blooms for a long time. And also the Ocean University of China and the First Institute of Oceanography, they're also involved in the studies of harmful algal blooms. In the southern part of China, we also have universities and institutes in Hangzhou, in Xiamen, and in Guangzhou to get involved. And so far, we have several key projects working on the mechanisms of harmful algal blooms, and also for the basic investigation of harmful algae and phytotoxins in China. So some of the results I will introduce later are actually products of these key projects. So I'd like to share with you some of the recent progresses of harmful algal blooms in coast waters of China. The first, for the toxins, are I think in the last decade we have increased understandings about the phytotoxins in the coast waters of China. Many groups in China are performed investigation of toxic algae and phytotoxins using metabolic coding, and using large volume sampling process to collect enough algae for the toxin analysis using LC mass and or HPLC. And then the toxins in different media like net concentrated phytoplankton, seawater, or sediment were analyzed. So based on this kind of investigation, we have identified several key regions with high risk of seafood contamination. For example, in the Bohai Sea, the coast waters of Qinhuangdao, in the northern Yellow Sea, in the area very close to Dalian, and in the estuary of Zhanjiang River, and also in Fujian province, in coast waters of Fujian province. So these regions are sometimes have high risk of seafood contamination, particularly paralytic shellfish toxins. And also there is increased understandings on the production of domoic acid producing diatoms. Because these diatoms which can produce domoic acid are not recognized until the year 2017. The first species recorded was Pseudoniscia simulans, and up to now there are 9 species which are capable of producing domoic acid were identified. And recently, during an investigation in the southern China Sea, we can see the toxins in seawater based on, in the net concentrated phytoplankton collected from seawater based on the crisis in this region. And another interesting issue is on the neurotoxin. So we can kind of BMAAs, amino acid, which can have a potential risk of human neurodegenerative diseases. And this toxin was usually considered produced by cyanobacteria in fresh water, but recently the group from the Ocean University of China, they identified these toxins from the diatoms in the seawater, and also based on the investigation of these toxins in Jiaozhou Bay, they found apparent biomagnification of these toxins. So that means the toxins can be concentrated from phytoplankton to high trophic level marine animals. And also we have more understandings on the toxins produced by the epiphyte dinoflagellase. So this kind of investigations was mainly carried out in the southern part of China. So they have identified several species which can produce different groups of potent neurotoxins. So that's some new findings about the phycotoxins in the coast waters of China. I think the results on domoic acid and also on BMAA are quite interesting, and this maybe could be included in the monitoring of toxins in other countries. And the second is about a very special type of harmful algal bloom which threats the mariculture industry. It was called brown ties caused by Orococcus endophiliferans. And this kind of harmful algal bloom was never recorded before in China until the year 2009. And in that year, we noticed a very large growth, the stop of growth of the small scallops in the coast waters near Qinhuangdao. And then when we start to look at the microalgae in seawater, we found this very tiny pelagophytes, Orococcus endophiliferans. And this bloom was continuously recorded in the following years until the year 2015. And then the scale or the biomass of the bloom started to decrease. And now there's no very serious problem of brown ties now. So to detect this very tiny microalgae, we developed high specific qPCR methods for the detection. And also based on the metagenomic analysis, we can identify that this species is a dominant species during the blooms. We had considered that this species maybe, the Orococcus endophiliferans may be transported from the other regions. It could be an invasive species because the scallop, which was very extensively cultivated in the coast water of China, was transplanted from the United States of America, where the brown ties is a very serious problem. But then in the following studies, we found that Orococcus endophiliferans is widely distributed in the coast waters of China, and also a group of scientists here in IOC found the DNA of Orococcus endophiliferans reserved in the sediment core, which can be traced a long time ago. So that means Orococcus endophiliferans is indigenous species. The optimal environmental condition promoted formation of this brown tide in Bohai Sea. The third type of bloom is microalgal blooms in the Yellow Sea. This also led to a very intensive negative impacts on the mariculture industry. For example, the accumulated green algae here in the coast of Shandong province killed a lot of cultivated animals, and also for this type of bloom, the scientists here has working for a long time, and the mechanism is almost found out. But the impacts of these microalgal blooms on marine ecosystem was not quite clear. So we tried to track the settlement region of floating green algae and found that most of the floating green algae were settled in the region southeast to Shandong Peninsula. And one very interesting thing is that we look at the phytoplankton communities after the settlement of green algae, and we found an apparent increase of pelagophytes, Orococcus, and Anopheles in this region. So there might be some linkage between the two different types of microalgal blooms. We have submitted these results to the Environmental Science and Technology, and hopefully it will be published. It could be published soon. And in the Yellow Sea, another type of microalgal blooms which threat the mariculture industry is caused by Sargassum hornbeam, which we call golden ties. So this intensive blooms of microalgae could damage the coral reefs of Popea, Puropea in the Subi shore. So in the year 2017, this kind of microalgal blooms led to a very serious damage to the industry. And the for Dunderfurnace bloom in the East China Sea, we also pose potent threats to the mariculture industry, particularly Karenia mikimotoi. So I'm very curious to know, the mechanism for the formation of a Karenia mikimotoi bloom, because for this type of bloom, they didn't appear every year like Dunderfurnace Crucentrum donghai. From the year 2002, there are only 2 bloom events in this region, and every time when Karenia mikimotoi form bloom, it will lead to huge mass mortality of cultured animals, either fish or shellfish. And the recent studies in this region found some new species. For example, the Calodinium diditatum. It also led to a very serious fish kill event in the year 2019. So that means in the region of coast waters near the East China Sea, there might be some new bloom causative species besides Karenia mikimotoi. And to make a short summary, I think this the Ocean Decade program offers a good chance for us to work together to do the monitoring and mitigation of harmful algal blooms. The studies I report this in the East HAB meeting. Our studies here in China has already goes from the phenomena observation to taxonomy biology and mechanisms of harmful algal blooms. Now I think we should focus more on the monitoring, prediction, prevention of harmful algal blooms to mitigate their negative impacts. So that's a short report. Thank you very much for your attention.
Thank you very much, Dr. Yu, for very comprehensive report from papers on your advancement of research in China. I see a hand from Nguyen. So yes, go ahead, Yuan.
Yes. What can I ask? What is the reason of reduce in frequency of red tide in China? It's because of you manage the nutrients better, or climate change, or some other organism competing with nutrients with microalgae. For example, you have inflation such as macroalgae bloom.
More than 0.1 cells, and they start to occur the PSP, measured by mass biopsy, and then when the PSP exceeded standard level, and we start to close the fish farms. And in Korea, the Alexandrium, the maximum cell density is about 10 or 50 cells per milliliter. And in this the PSP monitoring system, also there are several things newly updated. So the first thing is, in the past, we only used the microscope, and we knew the Alexandrium is the PSP species, but we didn't know exactly which species the PSP causing species. So we surveyed the last for the 10 years. So we tracked the Alexander species using qPCR, and we found out there are 4 major Alexander species in Korea. And that's the Catenella, Perspicum, and Fraterculus affine. And Catenella, that species occur only spring, and that's exactly matched to the PSP period. So that means the Catenella is a major PSP species. And for Perspicum, that occur every month, all season. So that means that this one is partly contribute to PSP. And the temperature range also exactly, the Catenella exactly matched to the PSP period. And each species has different temperature range. So that means these 4 species live in the same area, but they don't compete each other. So they have very distinct ecological positions, right? And also when the strong PSP occurred, the character is the Catenella cell density is higher and the temperature was low. That's our PSP pattern. And this last slide. This is this year PSP, our monitoring result. And the PSP that exceeded the standard level in the mid-February. And then the PSP peaked in the mid-April. And the red color is Catenella cell density. And that exactly matched to the PSP event. So Catenella is a PSP species. And then the Catenella decreased. In the meantime, the Perspicum increased. So that's why this year the PSP extended to the June. The one character of this year PSP was the PSP occurred the longer period because of the Perspicum occurred at high density in the June. So this is our PSP monitoring system. Thank you.
Thank you very much, Dr. Park. So I think we can have questions. Yes, Iwataki-san, go ahead.
Thank you. Good question. Why do you include the Alexandrium fraterculus for the PSP monitoring?
The reason, first, what we want to know is which one is the major Alexander species. So we measured all the Alexander species in Korea. So that's why fraterculus is non-toxic, of course. But yeah, we measured and then we specified which one is toxic or non-toxic.
Yeah, thank you.
Yeah, I guess I agree that it's always important to know what we have in our backyard, whether they are non-toxic and toxic, because they're so resemble to each other's under microscopy observations. So to able to tell that will help in the monitoring. So any other questions? I think we have to move on. We actually have roughly about 5 to 10 minutes left for our discussion. So if you have comment and questions, please raise your hand. So, and I guess we have a very fruitful and very interactive discussion today. So it's good to know that the emerging species that we have this region and how we can deal with that. So taxonomy is very important because correctly identify a species to species level and that relate to the toxic species. I guess that will be very, very useful for us to tell and very useful to the farmer and also to the monitoring agency. So of course, at the same time, we also have to learn to apply new techniques. It's good to see the very good progress on the prototype of monitoring environmental sensors that they're already in operations in Philippines, in Bolinao. So hopefully this will be translated into more area, more local government that will use the same technology for early warning, and we hope we also can use that. And it's good to see the helicopter monitoring and to early detect red tides in Korea. I guess that is also something, because most of the time the marine police will do their patrolling around the country. So that is some additional output that they can contribute to. And I also like the example from Wakita-san. So about this, how to use this business model to the toxic clam and not become non-toxic and become a business model in part of Japan. So I guess that is very, very useful, because when we look at the ocean decade outcome, we are trying to learn from each other's what are the practices that available around us, not only in our country but also in other neighboring country from our friend, that can be applied to adopt, and also for us to share with our local monitoring agency the possibility to adopt, like the apps. The Malaysian side also, we are trying to develop that to look at the Malaysian hub report. So but that is only focused on PSP. So I'm very impressed with the system already in operation in Korea, and also very comprehensive monitoring program in China. So I think that is it actually very costly. It's also something related to expenses. The damage to is something that we have to consider in safeguard public health, the operations or sustainability of marine cultures. So how to reduce back to the root of the problem is how we can condition nutrient discharge. So those are efforts that we have to look at. So maybe I'll go to Kita. So for you to say a few words or your comment.
Thank you very much, Potin, for sharing your comments. And I also would like to thank all of you presenters, good practices and very good knowledge development. And I would like to encourage all of us, including me myself, to do further collaboration in the coming years, because we are in the ocean decade and we have a very variety of opportunities probably in the coming years. So if you are interested in community collaborating with each other or some members among our members, and even to the members from the Global Hub, because Potin and Aleta is a member of the Global Hub project endorsed by the UN Ocean Decade, right? So
yeah. Yes.
Just thank you for all the contribution and all the participation. Any other comments from all of us?
Okay, so I focus and say you want to have
thank you very much. How to say, I congratulate everybody the progress of the hub, but one thing I wish to give one subject. Beside the face of reporting, you just left side have science for societal application. How to apply our knowledge, our science to society. That part, not much developed yet. So, for example, early warning, then the people have warning. For example, if typhoon come, people will close doors and windows and cover thick the material to prevent the house or anything. In case of HAB, if people get the monitoring result, how they can prepare, what they have to prepare, that kind of information, that kind of science, not yet developed well. Or in order to develop that science, we have to utilize WESTPAC or IOC system to collect all information. I give question to Wakita-san and Iwataki-san, whether we Japanese, whether we use knowledge which had from the Chatonella fish killing bloom, whether we apply that knowledge to Hokkaido Karenia red-tide cases occurring now. The answer is not yet. In order to that, we need to collect information, not only Japan, but also China, Korea, and Southeast Asia. And WESTPAC can provide the useful mechanism to collect that information. Thank you very much. I hope so.
Thank you, Sensei, for your very constructive comment. And I think that is very true. So I guess that is the differences or the shortcoming that we have to address. For example, the Alaska actually monitors citizen scientist monitoring program that help to monitor PSP in the region. So I guess similar, I mean, in our case, PSP are mainly the mandatory or the responsibility of the fishery agencies, and somehow the public are not directly involved. So I guess that is something lacking that in our side, that we can be more engaging. We should try to reach out to the fishermen community, fishermen associations, and even farm operator. I have been contacted by some farm operators, and they are keen, but the thing is, when talk about investment or initial capitals that related to how they can monitor their farm, so then they somehow they become a bit reluctant. So I guess that is something that we have to continue to approach farm operators so that they can protect their investment. So and for locals, I think I would like to also take the opportunity to share one, because of the lack of understanding by public. And now we have a lot of pseudo fake news or fake news that in the media, and that caused a lot of damage to our mariculture industry. For example, Sungai Geting alga bloom due to minutum and become the ban of shellfish for the whole east coast of Peninsula Malaysia that appear in the media. So because of a small bloom in an area that 2 or 3 kilometers affected very small, for small numbers of trader and harvester, become a warning to the whole east coast of Peninsula Malaysia that there are people are advised not to eat. I mean, from all these YouTubers and social medias. I guess that is also something that we have to carefully and look at in the futures, how to elevate the science awareness of public, and don't simply believe something, depend on the reliable sources of information. I guess what they have apps, authentic sources of information to the public, to the farmer, to the, so that will be, that will overcome this sort of problem of pseudo fake news or fake news in the social medias. So I think with the time constraint, we have to close our sessions. I would like to take the opportunity together with Dr. Wakita, so the convener for these sessions, and to thanks all of you for continuing to stay with us, for spending your time, Dr. Yu, Dr. Iwataki, Aleta. Thank you also to Fukuyo Sensei for coming over to join us. I'm very happy to see you, and Kinsun. So I think that is we have more opportunity to interact and exchange, certainly, and we will certainly keep in touch again from WESTPAC, from Global HAB, and of course on our personal capacity to exchange information that we hope that will help to minimize the impact, to help to improve the system that we have improve our understanding on HAB in the region for UN Ocean Decades. I think that is my closing. Back to you, Wakita-san. So
Thanks to you too. Thank you very much for TNA and all the speakers and all the participants. Thank you very much.
Thank you. Take care and stay safe. Bye-bye.
Yeah.
Thank you.
Thank you.
Thank you.
Thank you. Bye-bye.
Thank you, Riani. Thank you
very much. Thank you.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you.
Bye-bye.